السلام عليكم ورحمة الله وبركاته. آه كيفكم جميعا؟ من أنه العدد قليل، بظن أنه معظم الطلاب مش بحاجة لهذه المحاضرة، لكن إحنا مدام في اثنين ثلاث مش مشكلة بنشرح. so إحنا راح يكون نظامنا كيف؟ راح يكون في عنا المحاضرة، وأي سؤال يعني طرق في ذهن الطالب يرفع يده، وأثناء المحاضرة راح نجاوب عليها بإذن الله. راح أسوي عندك البرميشن عشان يسوي المايك اوبن عنده وبسأل السؤال عادي. بعد ما أخلص المحاضرة في يعني كم سؤال أنا حاطهم، راح نحلهم سوا، وهيك بظن إنه بكون شملنا المادة أغلب. إنه راح نبلش حاليا. اللهم صل على سيدنا محمد. بالنسبة للجلايكوليسيس، the glycolysis, it is the process in which the glucose is then converted to two molecules of pyruvate. And this is the process for splitting the glucose, okay, and splitting the sugar, because the glucose, it is a sugar and it is a carbohydrate. The glucose itself composed of 6 carbon atoms. عندنا الجلوكوز بيحتوي على 6 كربونات. ال6 كربونات هدول راح يتوزعوا على 2 molecules of pyruvate، لأنه كل molecule of pyruvate بيحتوي فقط على 3 كربونات. تمام؟ هاي بالنسبة للديفينيشن of the glucose. اللهم صل على سيدنا محمد. Okay. Wait a minute. In here, the glucose is that's the monosaccharide. monosaccharide اللي هي أبسط أنواع السكر, because we have kinds of sugars and أبسط نوع هو المونوساكرايد. الجلوكوز هو أحد المونوساكرايد. عندنا كثير في مونوساكرايد أنواع، لكن المطلوبات منا جلوكوز, fructose and mannose. This is three monosaccharides. Is that's called isomers. Isomers معناها إنه بيشبهوا بعض في عدد الكربونات وعدد الهيدروجين وعدد الأكسجين، بس بيختلفوا في الاستراكشر. So they are like same in the formula, but they difference in the structure. Okay, so you can see in here that the structure in here for the fructose is different from the glucose, but they have the same formula, اللي هي C6H12 and O6. I mean that the 6 carbon atom and 6 water molecules. Okay, the disaccharide is that two monosaccharide combined together by all glycosidic bond or any glycosidic bond. These bonds like attach one monosaccharide to another monosaccharide and make the disaccharide. Disaccharide is that two sugar units. في عندنا هون أحد أنواع الدisaccharides. Wait a minute, I'll remove that. We have in here some kinds of monosaccharides, and some like these kinds of monosaccharides will attach together to form the disaccharide. The disaccharide in here, the sucrose. بالنسبة لي أنا أحيانا الواحد بتلخبط في حفظ هاي المجموعات. ففي عندك for example the sucrose، طريقة حلوة تحفظها إنه ثاني حرف هو U. تمام؟ فاللي بيكونها لازم يكون عندك هون يو كمان هون يو، فهو جلوكوز وفركتوز ما في غيره. تمام؟ اللاكتوز في عندك ثاني حرف A وأول حرف L، فلازم يكون فيهم L الاثنين like جالاكتوز وجلوكوز. المالتوز from the same, from the glucose and the glucose. This you have to memorize it. أنا ما عم بظن إنه الدكتورة تركز على هاي الشغلات، لأن هاي الشغلات from the first semester. The polysaccharide, it is the polymers of monosaccharides. This is the definition of the polysaccharide. But if she asks you what is the monomer of the polysaccharide, هيكون الجواب monosaccharide. So the monosaccharide is the monomer, and the polymer is that polysaccharide. Polymer, polysaccharide, monomer, monosaccharides. Okay. Examples of the polysaccharides are the starch, the glycogen, and the cellulose. This is examples. The starch and the cellulose like are found in the plant, but the glycogen is the storage form of the glucose, and it is found in the animal, like human, animal, every animals. Wait a minute, I will say the number of students. Okay, everyone can hear me, yeah? Okay. And in here we have the transport of glucose. The transport of the glucose, it is like the method for transport the glucose molecule to like the tissues to the cells. And we have two methods. It's like sodium independent and sodium dependent. Like it's easy to understand it. sodium independent ما بتعتمد على الصوديوم، راح يكون بدنا طريقة ثانية اللي هي facilitated diffusion. And if someone remember from the first semester, we say about when you heard any word diffusion, like diffusion, simple diffusion, or facilitated diffusion, that's mean it is passive. So in here, we'll not use any ATP molecule, okay? So facilitated diffusion, sodium independent, we will not use any ATP, it will be passive. And we have uh, more transporter, like they are 14 transporter in the facilitated diffusion, but the doctor requires just 5. And here we have that's GLUT1. And why it is say GLUT? GLUT because it is that take the first three letters of the glucose, it will give you the glue. And T, that's mean transporter. So mean the glucose transporter 1, glucose transporter 2. And here you should like remember that GLUT1 is that used in the red blood cells and the GLUT transporter 2 in the liver, kidney, and pancreas beta cells. Okay, and GLUT3 in the neurons, and GLUT4 in the muscles, in the adipose tissue. And in here, focus with me a little, like GLUT5 we use it for fructose, not for glucose. It is right to say GLUT, glucose transporter, but this one, it is used for fructose. So it's that transport a fructose molecule in the testis and small intestine. Okay. Another information is not important, but you should like memorize the places and like GLUT5 for fructose, not for glucose. And it is passive. Anywhere you see GLUT1, GLUT2, GLUT4, etc., it is passive and it is facilitated diffusion. Another kind for transporter, it is sodium dependent. هاي الطريقة في النقل بتعتمد على الصوديوم. راح تلاقوا إنه الصوديوم والجلوكوز بيمشوا مع بعض. وين ما بروح الصوديوم بروح الجلوكوز. على هيك سميناها SGLT, sodium glucose transporter. Okay. This one is the non-passive. So energy requiring. نستخدم فيها الانرجي. نستخدم فيها الطاقة. And the movement of the glucose is coupled to the concentration gradient of the sodium. So where's the glucose is that's moved, the sodium also moved. And you can say, where's the sodium is that moved, the glucose also moved. Okay. In here, the definition of ATP, that's the ATP is that's the definition, adenosine triphosphate. That's energy, you know. But the energy, if she asks you where is the energy found, وين بتكون الانرجي موجودة؟ رح تجاوبوها انه في البوند، في الفوسفيت بوند. الفوسفيت بوند in here you found the energy. Energy مش موجودة في اللهم صل على محمد في الموليكule at all. Not energy not found in the molecule at all, that's found in the phosphate. In here you didn't find any energy. You can find it in here, in here, in here. Just in here, not in all molecule. The NADH, it is that nicotinamide adenine dinucleotide. And here you can like just memorize that NADH and NAD plus. What's the difference between them? That NAD plus it's need electrons to pH to give that hydrogen. So it's need electrons. So when that something need electrons, okay, is that we say accept the electrons. And the NADH is have electrons. She can give electrons, so it's donate electrons. And when it's give electrons, it's lose it. So is that happened oxidized. So the NADH, when it's lose the electrons, it will be oxidized, okay? And NAD plus, when it's gain electrons, it will be reduction process. Okay, now the glycolysis. As we know, like in biochemistry, we take about glycolysis, gluconeogenesis, and etc. And like each process happened in some place. And for final, we should like focus in which place that's happened. لازم تعرفوا المكان اللي بيصير في العملية، لأنه في عندكم أكثر من عملية وبتحدث في أكثر من مكان، وشوي الموضوع بلخبط. فعندكم أنتوا الجلايكوليسيس it's that occur in the cytosol at whole from the beginning until its end. It's occur in the cytosol. So as you see in here, the glucose composed of 6 carbon atom in that split and division, and we have a lot of serious reaction, then it's will become 2 molecules of pyruvate, and each one have 3 carbon atoms. This is a carbon, by the way. Okay, and here we have 2 phases, and in these 2 phases we have 10 steps. So these 2 phases that's divided to like some phase use energy and some phase generate energy. بعض الفيز بتصنع طاقة وبعضها بتستهلك الطاقة. رح نشوف كيف. And here, as you see, 2 phases for aerobic glycolysis. By the way, you should like focus at aerobic because we have without aerobic, non-aerobic. 2 phases for aerobic glycolysis, as you see, there's the energy investment, تستهلك, تستعمل, and energy generation, like تنتج طاقة. And here, like when the glucose is like splitting for the pyruvate, is that we'll use 2 ATP and we'll generate 4 ATP. لكن إذا طرحنا subtraction these ATP عشان ناخذ النت كامل, for take that the whole ATP like we produced in the end of this process, we will like understand that it is just will be at the net is that just 2 ATP. Okay, so that's important question. If she asks you what is the net aerobic glycolysis, it is that 2 ATP. That's the most important things. And 2 pyruvate and 2 NADH. Okay, let's see if there's someone have question. There's no one. Okay. And here you can see the steps and how it is that occur from the glucose, as you see in here, until the pyruvate, or another name is that's pyruvic acid. That's not important, but you didn't show to understand every steps. We just have maybe 4 to like memorize them, and another it is like just to see it. You will like forget it the next year. How to know that I should memorize that and I should not memorize it? See that this enzyme is important, and this enzyme is important. If like I lose that enzyme, the pyruvate kinase, I will have a disease. So I should memorize that. But this enzyme and this, if I lose them, I will not have any disease. So I shouldn't memorize them because they have no relationship with any disease. But I will explain everything, and you should memorize from glycose in here until here, or until here exactly, and these steps, phosphoenol pyruvate and pyruvic acid. And here the glucose pathway. First of all, it is the glucose. How it's that splitting? We know something called phosphorylation and dephosphorylation. فسفرة وإزالة الفسفرة. أول شيء في عندنا ال ATP ال ATP راح نستخدمه في أول steps. So the glucose, the first, we will use ATP. That's investment ATP. How? Like the ATP will come and the enzyme hexokinase, and we have another enzyme called glucokinase. Wait a minute. By the way, when that's the time end, we'll like make a new link, okay? So we have a glucose and the ATP will come. When the ATP come, we have enzyme called hexokinase. The hexokinase enzyme is that will take the phosphate group, and this phosphate group like will send it for the glucose and make it attach at the position 6, because we know that glucose, as you see in this photo, we have position 1, 2, 3, and like that. It will attach at position 6. Okay, so that's called glucose 6 phosphate. You can memorize it from its name. So the name tell you where is it attached and what's happened. So glucose 6 phosphate. So احنا أضفنا حبة فوسفات على البوزيشن 6. So glucose 6 phosphate. Okay, now the glucose 6 phosphate converted to the fructose 6 phosphate. And I told you that the fructose and glucose is just an isomer. It have the same formula, but difference in the structure. And the definition of isomers is that's important. Okay, the fructose 6 phosphate is have the same. Is that the phosphofructokinase, or PFK, as the doctors say, one, um, make this ATP, take one phosphate group from it, and send it for the fructose 6 phosphate and attach at the position 1. So is that called fructose 1.6 bisphosphate. And pi, that means we have 2. So fructose 1.6 bisphosphate. Then these things, the fructose 1.6 bisphosphate, will give me the dihydroxyacetone phosphate and also will give me the glyceraldehyde free phosphate. So until now I have one glyceraldehyde free phosphate and one dihydroxyacetone phosphate. Okay, this one, the dihydroxyacetone phosphate, will convert it to the glyceraldehyde free phosphate. So now I have two, not one. Okay, so after that, any step happened in here will be doubled. So then I will have 1.3 bisphosphate glyceride, 2. So like I will have two molecule of that, and two molecule of that, and two molecule of that. And anything it's generate, it will give me 2. So I will have in this step 2 ATP, not 1. And in this steps, I will have 2 ATP, not 1. So that will give me 4 ATP. And if this 4 ATP I divide it, or sorry, subtraction it from that 2 ATP in here, this will give me 2 at the net. Okay, so these steps in here, it's important for red blood cells. I will pack for it um, by just 10 minutes. But in here, come in here and focus about that. Phosphoenol pyruvate. The phosphoenol pyruvate will convert it to the pyruvate. هتتحول للبيروفيت عن طريق انزيم اسمه pyruvate kinase. The pyruvate kinase generation ATP, because can you see phosphoenol pyruvate? الاسم بيدلك انه هو المولكيول هذا يحتوي على فوسفات. فانا راح اخذ الفوسفات اللي هو محتويها واعطيها للاي دي بي، للادينوزين داي فوسفات، عشان تكون لي ATP. Okay, and give me the pyruvate, and it's 2 molecules of pyruvate because we have 2 molecules of phosphoenol pyruvate. Okay, wait a minute, I will see the time. Okay, it's 5 minutes. Like, I can continue. So in here, hexokinase and glucokinase, these molecules is that in the first step. I told you in here that we have the glucose, and the glucose that's converted to the glucose 6 phosphate by phosphorylation, the process called phosphorylation. So if she asks you about the process, how the glucose converted to the glucose 6-phosphate, you will say by phosphorylation اللي هي الفسفرة. Okay. And here we use two enzymes, and each enzymes have a special function. For example, the hexokinase function difference from the glucokinase. I will tell you the function, but just 1 minute. The glucose that's converted to the glucose 6-phosphate by these enzymes, and each one have like feedback inhibition. The feedback inhibition اللي هو عبارة عن مادة إذا زاد تركيزها تؤدي إلى إحباط أو توقيف عمل الإنزيم هذا. So the feedback inhibition is that an substance, if they have a high concentration, it will inhibit these enzymes. And they have different feedback inhibition, okay? As you see in here, that the hexokinase have different locations and glucokinase have different location. The location of the hexokinase will found in all the tissues. هو موجود في كل أنواع التيشوز، بس هذا لا يعني إني أنا بستخدمه في كل مرة، لأنه أنا في عندي تركيز معين من الجلوكوز، إذا كان زاد أو قل رح يختلف معي إني أستخدم الهكسوكينيز أو الجلوكوكينيز. الجلوكوكينيز that's found in the liver, pancreatic beta cells. If I told you that glucokinase found in the liver and pancreatic beta cells, you should know which type of transporter we should use. So I told you is that we have a lot of transporters, and is that some type of transporter used in the liver, kidney, and pancreatic beta cells. I will back for you. It is the GLUT2. So the GLUT2 will use it for the glucokinase because it is found in the liver and pancreatic beta cells, and also the glucokinase location in this place. Okay. What about the Km for the glucose? The Km we should like know what about it because we take it in the first semester. The Km is that it is that the concentration of the substance, and if this substance increase the concentration, this will make the enzyme work and be active to reach the half of the maximum velocity. يعني أنا في عندي المادة هاي اللي هي جلوكوز Km for the glucose. ال Km for the glucose هو تركيز الجلوكوز اللي راح يخلي الإنزيم active ويعمل ليوصل لي الماكسيمم velocity نصها. تمام، اللي هو زي ما انتم شايفين هون. فور اكزامبل عندي الجلوكوز كان تركيزها عالي راح يتم تنشيط الجلوكوكاينيز، لكن إذا كان تركيزها خفيف راح يتم تنشيط الهكسوكاينيز. So the Km is the concentration of the substance، تركيز المادة أو substrate which permits تسمح للإنزيم إنه يكون active حتى يسوي achieve، يعمل على إنه يوصل ويحقق ال half of maximum velocity. في علاقة عكسية وفي علاقة طردية مع الكي ام. الكي ام لما يكون لو راح يكون عندنا لو ماكسيمم فيلوسيتي لأنها علاقة طردية بيناتهم. Okay, so it is a direct proportional. But when we have inversely proportional علاقة عكسية مع الافينيتي مع الانجذاب. So the Km for the glucose in the hexokinase is that's low and low maximum velocity, but they have high affinity. يعني الهكسوكينيز لما يكون في عندي أنا جلوكوز بنسبة واطية قليلة راح يكون عنده هاي افينيتي، إنه الانجذاب الجلوكوز راح يكون كثير للهكسوكينيز حتى كانت نسبته قليلة. عشان هيك مهما كانت نسبته قليلة بنجذب بكمية كبيرة للهكسوكينيز وبخليه أكتف. But the glucokinase the opposite things، العكس، إذا كان نسبته كثيرة لأنه عنده high Km ولو سوري وهاي عندك maximum velocity راح يكون الافينيتي قليل، الانجذاب تبعه قليل. فإذا كان عندنا الجلوكوز نسبته عالية لازم يكون كثير concentration عشان يروح يشغل بيسوي active glucokinase. So I will send another link because there's less than 1 minute. Okay.